any questions that we get will be answered, and they'll be in a document that we will link to the courses and webinars page on our site. And I'll send an email out when all that stuff is ready. And the FTP site URL there, that compressed link, that will get you to materials, the notes for today's demonstrations, and the slides. Okay, so we're going to talk today about a program called QuickBLASTP, at least that's what we call it on the web. This is a program for rapidly finding high-scoring protein matches in large databases. My name, again, is Peter Cooper, and that's my email address if you have any questions about anything I'm talking about today. The credits for this, of course, go to Tom Madden and Greg Bortine from the BLAST team, and we're going to talk about some of the experiments they did with QuickBLASTP in just a few minutes. So we're going to talk about what it is. We're going to talk about why you might want to use it or why do you need it. Give a very brief, probably somewhat inaccurate description of the algorithm or the procedure that QuickBLASTP uses, but enough so you can sort of understand what it does. We'll look at some of the experiments that Tom did, looking at results from QuickBLASTP and comparing them to ordinary BLAST. And then we'll talk about how do you get the QuickBLASTP on the website, and also how to use the standalone binary that you can get to now as a demonstration. And then we'll hope to do some things live on the web and on the command line. So what is QuickBLASTP? This is a faster alternative for ordinary protein BLAST. It uses a specially pre-indexed database. We'll talk about that more in a few minutes. It's designed for rapid matching of queries in very similar database sequences. About 97% of the database sequences with 70% or more identity to the query are found, and about 98% with 80% or more identity to the query are found. This is compared to what you would get with ordinary BLASTP, so it's less sensitive. Why do we need this anyway? And there's a number of challenges that are really a result of the advent of high-throughput sequencing and rapid processing of genomes. The default BLAST database, which is called NR, now contains about 141 million sequences, actually more than that, and 51 billion letters. And its doubling time is about 2 years. So next year at this time, it should be over 100 billion letters. And many of you have probably experienced the fact that ordinary protein BLAST searches are taking longer than they used to. And if you have a highly conserved or highly represented protein, that means one there's lots of copies of it in the database or variations of it, a search like that can take now up to a few minutes. And of course, modern high-throughput projects are generating a lot of this data. At the same time, they have a need to analyze a lot of their protein coding regions. And really what you're trying to do in those cases, if you have a metagenomic sequence or a genome or something like that, you're trying to find the strong matches in the database, because many of those proteins will have close matches. So all this means that we need a process that will let you quickly identify strong matches to a set of query sequences. Sensitivity in this case is less important in most cases. So this is a very oversimplified description of what QuickBLASTP does. The first part of the QuickBLASTP search uses an alignment-free method. And basically it makes an index of the 5 mers of the database and the query sequences, and basically looks them up in memory to find candidate matches, which have good k-mer content compatibility. The second part is to take those candidate matches and do a BLAST alignment with them. And so doing that means you get to standard BLASTP alignments and statistics. The first part, finding your matches, uses that alignment-free method. So I'll just go over some results that Tom in the BLAST group presented at the NIH Research Festival recently. And they basically are the kinds of things we're talking about, where they looked at a proteome from Shigella flexneri, 4,799 proteins from that genome. This is a whole genome shotgun assembly project. And another kind of proteome, this is from a transcriptome shotgun assembly project. This is for a tick, Amblyomma sculptum, and 4,246 proteins. And basically, they did searches comparing those against NR, and they got some information that's very useful out of that about how BLASTP and kblastp perform. Kblastp is the name for the standalone program that corresponds to QuickBLAST. So for BLASTP, these two projects, the Shigella took 10 hours, and for Amblyomma, it took 18 hours. In the corresponding cases, BLASTP, QuickBLASTP, the faster method, reduces those times by a tremendous amount. So the speed up for the Shigella was about 13 times faster, and for the Amblyomma sculptum, it was about 20 times faster. So obviously this is a huge improvement in the speed at which you can analyze a set of proteins. This is looking at the number of hits found at various percent identities. Notice that as you go down in percent identity, kblastp or QuickBLASTP does a little bit worse compared to BLASTP. And so that little circle I have there is the 65% identity point. And that's a good point to say, you know, below that percent identity, QuickBLASTP is not as effective. And for this particular set of proteins, it's not quite as dramatic as it is for the next set. These are the ones from the tick. So you see that it departs fairly quickly after below 65% identity. And this has to do with basically what kinds of proteins are in the database and how closely related they are to the set of proteins you're working with. So you see less saturation of the BLASTP results with the tick proteins than you do with the Shigella proteins. Another way of looking at this is looking at the number of missed hits compared to BLASTP with a 65% identity cutoff and without one. And so you see fewer missed hits. This is the column here for 0 missed hits over here. There's a slight difference in the 65% bar there. It's quite a bit more difference if you look at that for the tick proteins. So the takeaway message from this is that this works well, but probably is only limited to things that are better than 65% identity, which is fine if what you're trying to do is to find what protein is this in the database. So how can you run QuickBLASTP or the standalone version kblastp? SmartBLAST, which has been around for a number of years, actually uses the QuickBLASTP algorithm already and has for several years. QuickBLASTP is now on the protein BLAST page as a program choice. I'll show you that in a minute. And kblastp is available as a standalone demonstration package on the BLAST FTP area, and I'll show you how to get to that too. So in terms of the live website, SmartBLAST, which is linked directly to the BLAST homepage and is also available and from your results, does use the QuickBLASTP algorithm. By the way, if you want to know more about SmartBLAST, we did do a webinar on that a while back, and that link there will take you to our YouTube channel, and you can take a look at that webinar. This is the standard protein BLAST page, and you can see that one of the program selection choices is now, and it's got the new icon there next to it on the bottom here, is QuickBLASTP. BLASTP is the default, but you can change this to QuickBLASTP to run QuickBLASTP. Right now, this is only available for the NR database, and that's because it requires a special index of those 5 mers of the database in order to run it. So we'd have to index the other databases in order to make them work with QuickBLASTP. You can also download a Linux package for this, quickblast.tar.gz, which is available in the demo area of the BLAST FTP area. There's a README there, and there's the tarball with QuickBLASTP and the indexing program as well. And if you get that README, it tells you exactly how to download and install this. It comes with several different programs, and I'll talk about what those are directly. This right now is only available for Linux. So what's in there? You get kblastp, which is the corresponding standalone version of QuickBLAST. It does the first part and the second part of the BLAST search. It's the search program. It has very similar options to BLASTP. There's the make kblast index. This is the program that makes the database index of the 5 mers. So it requires a formatted protein database as input, and then it's going to add some additional indices for the initial alignment-free search. The package also comes with the update BLASTDB Perl script. That's for downloading your BLAST databases. And there's also a sort of a script in there that does, it's called catvolumes.sh. What it does is it basically loads the BLAST database volumes into memory. You can sort of get a hot start on your BLAST search. That speeds up the search if you have a large database. The way this is written right now is it specifies the RefSeq protein database, but you can modify it to use other databases. So just a couple of things to think about. We've already sort of said this, but I just want to remind you again, QuickBLASTP rapidly finds closely related proteins. And so I would say that it's basically not terribly useful if your matches are below about 65% identity. We're going to show you an example of that in a few minutes. It shines if you're doing batches of queries. And so that's sort of the idea behind it, is that you're analyzing a large number of protein sequences or a smaller number on the web, but still several of them. The standalone program, for this to work, it requires that the BLAST database plus the indices be able to be completely loaded into the RAM. And just sort of a note, if you're thinking about the default protein database, which of course is very large, the NR database plus these indices require between 200 to 250 gigabytes of RAM. The index files for kblastp occupy more than half of this total. So you need a machine with lots of available RAM in order to do this effectively. Okay, so I'm going to leave the slideshow now and do a couple of demos, one on the web, show you what happens when you do protein blast and QuickBLASTP with the same set of proteins and the same settings. I'm going to take 5 proteins from that Shigella flexneri proteome that I talked about a few minutes ago. And then I'll show you a problem with sensitivity that is one we already talked about, but just so you can see what it looks like. I took some of those tick proteins which have less good matches in the database, and you can see what happens when you get below about 50% identity. And then we're going to go over to the command line and do some kblastp searches with 33 proteins from a plasmid from Serratia liquefaciens. I'm going to do 2 things here. We're going to use the BLAST interface here. Let me make that a little bit bigger so we can see it. And I'm going to use these notes. These are the notes for my demos, and they're available, by the way, on that FTP site. So if you ever want to do these things again, and the RIDs have been preserved, so the links to the searches will work. So I'm just going to put that in your face for a moment. And I'm going to go ahead and just show you the speed up, hopefully, with these 5 Shigella proteins. So I'm going to select them here. I've got them in my little notepad here. Copy them. And I'm going to go over to the Protein Blast search page here and paste them in. Okay. So they're ready to go. And now by default, the settings here are to run BLASTP. So what I'm going to do is I'm going to start that BLASTP search running. I'm going to open my results in a new tab here. And I'm just going to change a couple of things. I'm going to set this to get 500 hits, just a couple of things that people would typically want to do here, and set the e-value cutoff to 10 to the minus 6. And let me go ahead and start that running. So it's loading in this tab over here. And this is my BLASTP search. Let's go back. I'm going back here, and now I'm going to change this to QuickBLASTP. And it changed my max target sequences. I'll go ahead and set those to 500 again, so it's exactly the same as the settings we used for BLASTP. And I'm going to go ahead and run that now. This is where it gets interesting. Let's hope this works. So what we can basically do is watch the time since submission. And my experience with this is it takes about 10 seconds. Let's see if that's true. Yeah, pretty much. It takes about 10 seconds. Meanwhile, the BLASTP search is still running. And with an organism like Shigella, there are lots and lots of proteins in the database that are very similar to the ones we're working with. And so this search worked pretty well. You can go through here and look at your results. Each one of your query sequences got lots of hits. Here's one that didn't get a lot of hits. So if we look at the scores down here, I hit a Shigella protein. It's got a pretty good coverage, and it's 100% identical. I hit the same protein, essentially. It warns me about that. It says your search has less than 10 matches. You may want to run the search with BLASTP. In this particular case, though, this is probably the right answer, but you could go back and try it again. I'm going to show you a case now where the result you get with the QuickBLASTP is probably not the right answer. So by the way, my BLASTP results did come in for this. So I can take a look at the 98 one here. And really, I did get one other hit, but it's a fairly weaker one. So this one's probably okay. There is a hit at 51% identity that I get with BLASTP that I don't get with the QuickBLAST. Now let me show you another example where you have to watch out. So, and that's when you get down into low percent identities. And this is from the Embryoma Sculptum transcriptome. So I'll get the QuickBLASTP results here. And I'm just going to open that into one of these tabs here. So these were 4 proteins. I can just show you what they were here. So I just got those from that transcriptome and I ran the BLAST search with them. First one looks fine. There's lots of good matches. Just let me show you this last protein. So it finds 1 hit. And notice that my percent identity is very low, 27%. So this is something that I just basically wouldn't want to think about in terms of using this as a result because it's too low for QuickBLASTP to be useful. Just to show you the difference, I'm going to go ahead and load the BLASTP results so you can see what happens with those. I'm going to copy that. I'll paste it in the same window. And so let's look at that last protein again. Remember, this protein found that match in kblastp was 1 match. Actually, this finds lots of good matches that are highly significant when you're using BLASTP. So just be careful when you're looking at a low percent identity. You don't really want to be using the QuickBLASTP, but it works great if your sequences are very similar to each other. Okay, or they have lots of examples of themselves in the database is maybe another way of putting that. Okay, the next thing I want to do is to go and just show you the standalone package. And if you go to the notes here, it's got the setup commands and everything, and I've already done those. So I downloaded QuickBLASTP, I untarred it, and I downloaded the SwissProt database because that's a small database and we can run searches fairly fast. This is the command that you would use to index your SwissProt database, and I'm not going to run that for you live because it takes a couple of minutes, and that's kind of a long pause. So let's go ahead and we're going to do a search. And just as a bonus example, I'm going to show you how to get use E-link to get the protein sequences for this Serratia plasmid. So let me just put that command in. This just basically says I'm going to get the accession of that plasmid from the nucleotide database. I'm going to go to the protein database and fetch the FASTA proteins for this. So let's do that first. And I'm going to go over here to a command window here. Okay, so let's run that. That shouldn't take but a few seconds. Okay, so let's just make sure that that really worked. So I have a bunch of proteins from this Serratia liquefaciens plasmid. And now I've got a couple of BLAST command lines here, one for kblastp and one for BLASTP. And I'm going to run them under the Unix command time so we can see how long they take. So here's kblastp command. And this command line, it wrapped a little bit, but that's okay. This is really, the options are no different than they would be for ordinary BLASTP. So I've selected my database, SwissProt. Here's my query, which is that FASTA file. I've set an e-value cutoff of 10 to the minus 6. And I'm using one of the tab delimited output formats, output format 7, and then I'm just going to make an output file here. So time is going to tell me how long it took to run this. And it usually takes about a little bit under 10 seconds. Let's see how it goes. And of course, how long it takes is going to depend a lot upon the machine you're using and what's going on that machine and things like that. So the real time here was about 9 seconds. And let's make sure that that wasn't a trick and we didn't get anything. Maybe we got nothing. So let's check it. So let's go, let's look at that output file. So we got lots and lots of stuff here. So this tab delimited format, these hit lots of SwissProt records. There's a big file there. And I'll go ahead and start the BLASTP running. Same query, same command line basically, but a different program. And that will run for a little while. So while we're doing that, I'll go back out to the slides and just point out where you can get some additional information about BLAST and other parts of NCBI as well. So let's go ahead and run that. So just a reminder, the BLAST homepage is there. It has its own special URL, blast.ncbi.nlm.nih.gov. And there's a little help link at the top. That takes you to a nice page that has lots of documentation available there. And there's a news feed. There's also a mailing list you can subscribe to. I'll show you the list, how to get to that in a minute. Also, a reminder that Tao and I sit on the BLAST help email alias, and you can write to us anytime you have a question about BLAST. As I often like to say, it's a good idea to send us your RID if you have a problem with a BLAST search so we can see what happened. Additional useful links here. There is a BLAST help book on the bookshelf that has several different parts to it, including how to set up BLAST, the command line standalone manual. That's how to get to the mailman list for BLAST Announce. And there's some general things that you should know about. We have a blog, NCBI Insights. We have a YouTube channel. We also have a support center, which is a place where you can go. If you have a question, you can go there and you might be able to find the answer to your question. If you can't find the answer, you can send us an email, and that can be about BLAST or any of the other NCBI services. There's also a set of fact sheets on our FTP site. Many of them are about BLAST. Tao is the major contributor to that. So there are lots of things there about how to set up BLAST. So let's escape from here real quick and see if that finished. No, it's still running. We can check back in a few minutes. Nope, just finished. So it took about 2 minutes to run that. Okay, so that's pretty much all I had to talk about today. So I'll throw it open for questions if anybody has any questions. If you have a question about the webinar program in general, please write to webinars@ncbi.nlm.nih.gov. Thanks everybody for coming, and we'll see you next time.